I'm Tyler Johnson. I'm a graduate student in oceanography at the University of California, Santa Cruz. And how this whole situation began started with the realization from a colleague of ours who did some diving out here on the lake and came across a sponge in Lake Tahoe that had never been reported before. And why that was interesting is that Lake Tahoe is a classic oligotrophic environment, meaning that it's really low nutrients. There's very little nutrients in the water. There's very little nutrients in the surrounding soil or on the lake. We're surrounded by mostly these granite rocks. The bottom of the lake is mostly sandy, so organisms that make up this lake, such as the rainbow trout and the mackinaw and the crayfish, have to really compete for nutrients and food sources in this environment. And why that's of interest for us is that the ubiquitous crayfish in this lake have been known to even eat the paint off the bottom of the hulls of boats around here. But what we ended up finding is that this sponge was not being predated or consumed or grazed upon by these crayfish whatsoever. And our suspicion was that maybe there's some kind of chemistry within the sponge that's deterring predation, because the sponge would be a classic source of nutrients for either the fish or the crayfish on this lake, high sources of protein and fat, but yet it's not being consumed. So our impetus behind this whole research weekend was to kind of go out, locate this sponge, collect it, and then bring it back to our laboratory in Santa Cruz to see if we can probe for any interesting chemistry in there that's serving to deter predation of the sponge. See, because the sponges, they don't have any forms of defense other than the chemistry they can produce to deter predation. They can't swim away. They're sessile organisms, so they've adapted in other parts of the ocean, for example, these interesting molecules that deter predation from other fish and invertebrates in the sea. And that's ultimately what leads our research group as it is today. We have a strong research program based around Indo-Pacific marine sponges, where we go out, make field collections of numerous different species of marine sponges, and bring them back to our research lab to isolate different molecules for biomedical research. And the hope is that this Tahoe sponge might have some other interesting chemistry. And we were successful this weekend in going out, locating the sponge right off the point down there in Glenbrook, Dead Man's Point, bringing it back, and we've collected it, and now we hope to see if there's any interesting chemistry that emerges in the coming weeks. And this was all